Then we have the alkaline phosphatase. This alkaline phosphatase enzyme, this removes terminal phosphate group, that is at 5' and the phosphate group is present, it will remove the terminal phosphate group. So what we basically do, we use the radioactive labeling so that we can radio label the phosphate with the p32. So this strand, this double strand DNA, when we use the alkaline phosphatase, so this will remove the phosphate group at the 5' end. So we have here the phosphate group. So this phosphate group is actually removed by the alkaline phosphatase by hydrolytic cleavage. So it is replaced then with the radiolabeled phosphate. So when we use the radiolabeled phosphate, so that the normal phosphate will be replaced with the radiolabeled phosphate. Then we have the polynucleotide kinase. This polynucleotide kinase, it transfers or it adds phosphate from ATP to 5' group of dephosphorylated DNA or RNA. So this is the dephosphorylated DNA. So we use the radiolabeled phosphate here, that is the p32 from ATP, to add the phosphate group at the dephosphorylated 5' OH group. So in this way we add the phosphate group at the dephosphorylated 5' OH group. Then we have the terminal nucleotidyl transferase. So this terminal nucleotidyl transferase, this is the enzyme that adds the nucleotides to 3' OH group of a DNA fragment. So this will add the this will add the nucleotides to 3' OH group of a DNA fragment, and this is used in homopolymer sticky or cohesive tails at 3' end of DNA fragment, and it helps in joining fragments with blunt ends. So when the blunt ends are present, so that it will help them to join. So this is used to make radioactive DNA probes by end labeling. So when we have here the poly A tailing, then terminal nucleotidyl transferase comes and it adds the nucleotides at 3' OH.